can. So 1220 to 130 today, 135 Emerson, just down the hall. Neat. I'm looking forward to hearing what they're going to say. Okay, before I go into the PowerPoint presentation, I want to show you a short video clip, if it works, of a number of years back, the Discovery Channel up in Canada, when I was teaching up there, did a piece on some of the work we were doing on common ragweed. And we were looking at a biological control agent, okay? A beetle, and actually two beetles that were consuming, or at least we were hoping would be a good way to control the common ragweed. Now, this is not classical control or inoculative control. Why? What I'm going to be talking about. Why? What would be the first thing you say can't be classical control?
You're dealing with something in its own range.
Right.
Like go back to where it came from and find something and bring it in.
Ambrosia artemisifolia, common ragweed, is native to North America. And so were these beetles. So in a sense, what we were doing was what we call conservation or augmentation biocontrol. Okay? So take a look at the report. It's 10, 15 minutes. And anybody who suffers from hay fever is going to relate to this. Take a look at the hair, a little darker than it is now. So this would have been probably about 7 years ago. 7, 8 years ago. Yeah, my last year in Canada. So, yeah, John Losey showed this video from Frontline, and he looked like he was a pretty young fella there. Let me make sure that I put this. Where did I put it? Put this in the right. Okay. That's what it is.
But now it looks like ragweed may have finally met its match in Montreal lab. You're looking at a statistic. One of the 500,000 workdays lost each year thanks to allergies. This chair's usual occupant is Jennifer Tack.
Well, the itching, burning, and redness of my eyes get so bad that people actually tell me that it looks very painful and that I should go home to take care of them.
1 in 4 Canadians has allergies. Symptoms range from a few sniffles to life-threatening asthma. Like Jennifer Tack, most sufferers complain this: Ambrosia artemisifolia, common ragweed. As a botanical adversary, it's nothing to sneeze at. So at McGill University in Montreal, they're on the attack.
The male part of the plant produces the pollen that causes the allergy. And here we have the female part, the location where the seeds are produced.
Those seeds are ragweed's defense mechanism. Each plant produces 3,000 to 5,000 of them during its seasonal lifespan, but not all germinate the next year.
They will remain dormant in the soil for, in some cases, 15 to 20 years. So if we've done a great job of controlling these plants for 15 years, but we missed that 1 year where 1 or 2 of them escape and are able to reproduce and start the cycle is renewed, and we get 3,000 to 5,000 seeds back into the soil. So you can see why it's a problem.
The other problem is that ragweed is incredibly opportunistic. It not only grows, but thrives in disturbed soil. Land that's been tilled or bulldozed, roadsides ravaged by mowers and snowblowers.
It's known as a pioneer species. It's one of the first plants to move in to areas that have been denuded of vegetation. So it just has these great characteristics of survival and being able to grow rapidly and reproduce. And so, yes, it's a terrible weed in a sense, but a great plant from an evolutionary perspective.
And in the weed control war, ragweed has certainly had the upper lead. It's grown resistant to most chemical herbicides. Even when you mow it down, it grows back to produce seeds and pollinate. Only one method really gets rid of the weed, yanking it out, roots and all, one plant at a time. But now scientists have found a better way by examining ragweed's roots, the historical ones, that is. Unlike most weeds which originated in Europe or Asia, ragweed is a native plant. That means it has natural enemies here.
How are ragweed plants doing?
Much better than because.
At McGill, entomologists Ina and Myron Teschler have enlisted 2 of those enemies, 2 species of beetles native to North America. Zygogramma saturalis, the big guy on top, and Ophrayella communa. No one's sure why, but these insects' entire life cycle revolves around ragweed. They feed on it, lay their eggs on it, then the larvae hatch and feed on it. Boy, can those bugs
insects were applied on those plants. And we have a treatment where insects were applied on the same age of ragweed plant. And as you can see, the ragweed plants are completely destroyed, they dry, they damage, and this happened during 2 weeks.
And these beetles have such discriminating taste, they'll starve to death before eating anything but ragweed.
Here we see the lettuce planted in the flat, and the same amount of insect was released in the cage. You see the lettuce is healthy, no damage on the leaves, and no dead plants. At the same time, you can see the dead insects, the dead beetles here. They were starving, and without the food, they eventually die.
Now, why is it that these beetles are in North America and we still have large ragweed populations? Of course, a question that we ask ourselves. But one of the answers is that basically they have natural enemies, parasites, that keep their natural populations down. So the goal here from our research group at McGill is to try to mass rear these insects under favorable conditions in the laboratory or in the greenhouse, and then at the appropriate time to release these insects in the field as adults. And then they would reproduce on these plants and keep the population relatively at low thresholds.
Using nature to combat nature is known as biocontrol. And this may turn out to be the weapon against ragweed allergy. So it may seem odd that it's a farmers group that's funding the McGill research. But you only have to look at Michel Legault's onion field to see why. This ragweed infestation will cost him $30,000. He's hand weeding only where the crop is salvageable.
The onions have been, we get small onions like this. They're not commercial, they get too small, they just throw them out. So this is where, in this area, we get down, and it's no use trying to get the ragweed out. It's too much. It's too late.
At least here, the ragweed is easy to spot. In carrot fields, it's next to impossible to tell carrot leaves and ragweed leaves apart. Many farmers are so frustrated they've resorted to spraying herbicide illegal for use on vegetables to burn the ragweed leaves a darker color. This agronomist says hand weeding costs farmers a fortune, not just in labor, but in losses as well.
Ragweed make a lot of root, so they will pull some carrots with ragweed, so that's lost. There's also some carrots that will be exposed to the sun, so all those carrots are also lost, because they will turn green at the top here, and it's also lost.
Before the scientists can send beetles to the rescue, they have to make sure the insects won't harm the crops.
As you can see on the flat where we didn't apply any insects, you could see lettuce, onion, carrot plants, and ragweed plants as well. And in the flat where we apply the insects, you can still see carrot, onion, and lettuce, healthy plants, no damaged leaves, and the ragweed plants, which were completely destroyed.
Pretty solid evidence, but the true test is in the field.
Ragweed is blooming right now, and it produces a lot of pollen, and Myron is suffering from the allergy from the ragweed. So that's why he's not here today.
The project started this year from scratch. Scientists had to wait until spring when there were beetles and plants to bring to the lab for breeding. In July, when they had enough beetles to release, they thought it was too late to have an effect. They were wrong.
We know that those insects work on the seedlings and when the plants are in the young stage, and we really don't expect those results on the big plants like this. But as you can see, the plants are completely defoliated. Yeah, this is okay. This is fresh, so we can use it.
Now comes the fine-tuning, figuring out optimum temperatures and lighting conditions for beetle breeding and ragweed growing. Because to breed these bugs in the lab, you need a year-round supply of ragweed. And ironically, the plant that manages to grow everywhere we don't want it to doesn't grow easily in the lab.
We typically have a hard time to get these to germinate. If we're ever thinking of bringing this to a production level or a commercial level, we need to get a better understanding of the kind of media that is going to be used to rear these insects. We can't simply constantly provide them ragweed plants.
So they're looking for alternatives, like a ragweed extract produced in the lab. To breed beetles year-round, they'll also have to figure out how to speed up their diapause, the insect version of hibernation. It can last up to 9 months. The scientists are so confident they'll work out the problems, they're already looking ahead to the next phase, teaming up the beetles with a ragweed-attacking fungus discovered by another McGill researcher. The beetles would chew on the plant, creating a port of entry for the fungus, a biological 1-2 punch.
I think it would be a little utopian to think that we'll eradicate it. Certainly, I think the goal and the goal and strategy is to try to bring it to low enough levels where, yes, it could still be there, and I'm sure it's still going to be there, but like I said, that we can tolerate it.
Some experts estimate that we'll have to reduce ragweed pollen by 80% for people with allergies. But overkill could throw off nature's balance. Without enough ragweed, these beetles would become extinct. And right now, they're probably our best bet against a stubborn and prolific weed that brings tears to the eyes of allergy sufferers and farmers alike.
Now, there's a nice crop, though. You should have
When
seen
we come
these.
back, how hurricanes go.
Okay. What's for lunch today? Okay. Yeah, that grower lost, $30,000. The year after, I think he was down to $15,000. In terms of an update on that project, after I left McGill, remember, this was a vegetable cooperative producers that paid for the research. And they did come up with a diet for the, well, I guess a diet, an artificial diet for the beetle. The larger beetle, the Zygogramma, turned out that it was okay, but it didn't have high fecundity, enough fecundity to make it worthwhile. But the little guy, the Alpharella, was really good. It was able to lay eggs, a lot of eggs, females did, and both the adults and the immature stages consumed the leaves. So right now, I think where the technology is that the actual cooperative of growers, they themselves, we passed on this technology to them, and they're working on, in individual fields and individual counties in Quebec, releasing some of these, some of the insects, and they've had some pretty good success. So a couple of things to keep in mind. You heard the story about, the growers, and I saw this firsthand. They were using herbicides illegal for use in vegetables in their carrots. They have onions, carrots, and lettuce. That's why those were the main species we were. These are muck soils, just like up in Batavia area. Really kind of dark, high organic matter soils. And they were, using these herbicides just to burn the ragweed so that even for hemp picking, you know where it was. Otherwise, as you all know, they were pulling out everything. And even with this, stuff was coming out. It was just horrendous. So that was a real concern because we didn't, these products have not been approved in vegetables. So as soon as kind of the equivalent of the DEC and so forth, was trying to find out, then they had to basically stop. So it's been relatively successful. It hasn't been as successful as we would have hoped. And why is that? What do you think? Sorry? Winter. But what is the reason why this already starting? These are both here in North America. Remember I asked the question, I said, geez, if these insects are so good, why is it that we have ragweed everywhere? And what was one of the parasites? Natural enemies. I mean, that's that balance, right? So one of the issues that's come up is, what happens when you mass produce these and just release them in these fields? I mean, then are you putting things out of balance? And that certainly was something we were monitoring. I mean, we weren't just going to be releasing them, but they're doing it at a smaller scale now. These insects don't move that far. I mean, it's not like it's both good thing, bad thing. But it's where they have high densities that they're using it. The vegetable farmers, the last I heard. So they were mass rearing themselves. They got over this diapause issue. They were able to get the diapause fairly quickly, and they would release the adults in the fields in early, late spring. So it would be probably May, early June. They did a number on the plants, but they feed exclusively. They don't touch anything else. Remember, they showed they died. That's amazing. Now, in time, could they move somewhere on something? Who knows? But boy, it's been millions of years they've co-evolved with. ragweed. Any questions? Yes, ma'am.
When you're doing the mass rearing, could you like put the pathogen, like the parasites that attack them, so that you could breed a group of these beetles that wasn't susceptible to their natural parasites?
Oh, and well, you could, but it's not just natural. You know, it's not just these parasites. It's not internal. A lot of them are what, you know, a couple of them, for those of you, were stink bugs that would go and get, when we'd have our immature stages, they'd just go and pierce them, you know, and just on the leaf and knock the beneficial inside. So how do you protect them from those? And you can't go in with an insecticide; you'll kill everything. And that was the other issue too. How, you know, this wasn't. In some cases, these weren't just organic. These were conventional growers, and they would still be going in and putting in a fungicide, and sometimes they were trying to, you know, put an insecticide. And so you really have to work with them and say, well, hey, you know, if we're going to release these insects, you got to be careful. You can't go in and start putting tefluthrin or, you know, or some other, you know, general insecticide. So it's tough, but a lot of them saw the value. I mean, it worked. I mean, it saved them a lot of money going in there doing this. But this would be, and I'll talk about, this would be kind of a niche market. But, you know, vegetable production in this area is big time. I mean, this is like up here. Austin.
If it's a natural species like this, can you release as many as you want without having to go through the process of getting them approved?
Yep. We didn't need any special permits. We didn't have to go to APHIS. That's the nice thing about it. The fungus was one of our graduate students in our lab that found the fungus attacking it. And so, yeah, that's why I was saying, you know, classical control. But the issue with using, you know, native species, in both cases, both the plant and is this whole question, are we going to create an imbalance and shift the ecological equilibrium because we're going in and dumping, you know, 2,000 of these insects that, you know, of course what we saw was, yeah, we dumped these 2,000, but guess what happened? Just like that graph, the parasites and predators also went up. And when they went down, they would go. So nature has a way to kind of keep things in check. But at least short term, when we did use them, so this is not going to be a self-sustaining. That's what we realized. It's not going to be self-sustaining. These growers would need to still do some inoculation, i.e. every spring, and in fields that were going to be particularly bad, they could release these insects. And yes, they would calculate what percentage they'd lose to parasites and so forth, but kind of rotate, move it around so it's not always the same, so the predators don't just, because the predators will, you know, basically increase to the point where it's not worth it anymore economically. I wish I should have, maybe I'm trying to invite one of my colleagues on this project to come and talk to us and update on, you know, here in CSS, but if ever you see it, you might kind of remember this. Okay? So, just kind of telling you, so it's biocontrol, but using a native species to control a native, you know, plant, like ragweed. Okay? So, let's talk about some classical control here. We had started talking about that whole, can you guys see? If not, we can turn another light on. But we were talking about in Queensland, where there were 35 million acres infested with this prickly pear, Opuntia inermis. And then, you know, this release of this moth, you know, within 7 years, just really, and this is a picture taken at the same location, and it's done, you know, wonders. We talked about that. This cactoblastis moth that really did a really tremendous, well, it was tremendously efficacious and also allowed pathogens to get in. So the larvae of the moth would feed into the ear, you know, the ear pads, as we call them, and then the fungus would be able to move in. A bit what we were talking about in that video, as a 1-2 punch type thing. Yes, I mean.
Is it 60 million acres or 25? Because in our notes it says 25.
Geez, did I have 25?
60, you changed it. You had 25 last time, I think.
Oh, okay. I'll double check. I mean, I should give you the correct one. I have it. I think it's.
It says 25 million hectares.
Oh, hectares. Hectares. So multiply by 2.5, 2.48, and it might be. Okay. Just a couple of other examples of classical biocontrol. You know, generally these have been fairly successful. Remember, these are non-native species that have been brought over from elsewhere, and we've gone back to the country of origin to try to control. This is, if you go down to Florida, you'll see a lot of this stuff. Pretty plant, water hyacinth, okay? That was one of the questions. Give me an aquatic weed. A good number of you picked this guy. Some of you water milfoil. Okay. So this is Eichhornia and Neocatina. Okay, it's really neat weevil. The mottled water hyacinth weevil has done a really tremendous job. And here's the damage that the larvae do. And again, there's a 1-2 punch situation here. They've also introduced a pathogen. So they have the weevil go in, create that first point of entry, and then the pathogen. They've got the pathogen going. A lot of work. Any of you interested in kind of biocontrol, aquatic systems? Florida's the place. I've got some good colleagues there, always looking for good grad students. Okay? And of course, that's such a big issue down in Florida. But this is another good example of a pretty good success story. It's still ongoing, but still, there's still some work. Chrysalina, okay, was used to control Hypericum perforatum. Do you guys know what the common name is? St. John's wort. Okay, Hypericum, the mood-enhancing yellow-flowered plant that you often will see. It's been successful in some parts of the country, particularly in the Northwest and so forth, but in other places it hasn't worked very much. And I just wanted to show you this example that sometimes it's regionally things seem to work. We don't know if it's the climate or what it is why in some places it's successful, but in others it hasn't been. And so there's continuing work in this area. Something closer to home, purple loosestrife. A lot of you are familiar, kind of knocking off our cattails. Okay, here's a picture I took in one of the water. This used to be a pond, I think, and it just dried to complete. You could walk right through this place. And some of the work that's actually been done here at Cornell by Byrne Blasi over in Natural Resources, using some real good success using Gallericella. If some of you have gone up to, I'm trying to think of the Montezuma, okay? That place used to be just covered with loosestrife. And if you go now, after the release of these insects, particularly the larvae that just browse even the insect, it's a lot of native species have come back. You go up to that area, you'll find some loosestrife, but it's here and there. We don't have those patches. Again, in some areas it's worked really well, in others it hasn't. One of the issues with these beetles is that their life cycle doesn't necessarily match when the maximum growth of the plant is, in the sense that these insects are feeding in June, July, but then their life cycle kind of ends. They go into the ground and so forth, okay, kind of lay eggs, pupate. And at that time, if the plant hasn't been totally defoliated, it still has time in the month of August to grow back. And so they're looking at maybe using a combination, maybe a root feeder and a leaf feeder. So that's also the approach now, is to kind of think about how can we attack the plant in its susceptible spots. From an ecology, biology perspective, the kind of stuff I do is trying to figure out what is the weak link in the life cycle of the plant. No use releasing an insect that feeds on seeds when the plant is now limited by seeds. Okay, like this would be a good example. You want to go after a part that's, is it going to be the root? Is it going to be the leaf? Is it when the plant goes from a seedling to a vegetative plant? Where is that transition? And you can actually do some really nice basic ecological research to answer those questions, such that then the biocontrol practitioner can better target. So you're not just blindly saying, I'm going to release this insect, we think it feeds, and let's see what happens. You have a good idea. Okay, the weak link is from seedling to first-year vegetative stage. Let's go get it. And that's, for example, that's how they've targeted garlic mustard. The transition between, garlic mustard is a biannual plant, right? It's first year, it's this little rosette, and then the second year it bolts. They've targeted the insects here at Cornell, researchers here, at the rosette stage, that first stage. That seems to be the real weak link in that plant, okay? So think of that, and this is, like I said, this is another of the success stories, okay? Did not want to leave you with the impression that for classical biocontrol or inoculative control, that's the same, remember that terminology, okay? That it's limited to insects. By far, the most widely used control agents are insects. There's no question about it. But we have used some rusts, some fungi. These are rusts, for example, to control something called skeleton weed in Australia. Okay? Very host-specific relationship. If there's actually different isolates of the rust attack different biotypes of this skeleton weed. I think we do have some skeleton weed down in Arkansas, in those areas. Okay, so, but it's not as problematic as in Australia. Yellow star thistle, big issue. Anybody from the Southwest here or knows? Star thistle is like the #1 weed in the West. You go to California, okay? Centuria, so very closely related to our spotted knapweed. Okay, Centuria maculosa was one of the plants you had on the Weed ID practicum. Okay, and they've got the USDA ARS group released this Puccinia, which is a rust that's very specific and has done a really good job. They released it in 2003, and it's really targeting this yellow star thistle. This thing is bad news. It's one of the main reasons there are also a lot of fires. It invades areas and is very fire prone. It knocks off natives that are more fire resistant, and it occupies these. So when you get these fires from lightning, that's why you hear sometimes out west what's going on. Part of it is invasive plants have knocked off natives that were fire resistant, and these guys burn like there's no tomorrow. Just okay, in the Asteraceae family, right? The composite. So that's one. And musk thistle they've used this Puccinia to control the musk thistle, which is a problem weed. There have been some concerns with the pathogen perhaps moving on, or the rust, on some native thistles. And that's something that you always have to be careful. But it doesn't seem that that's a big issue. We have a lot of native thistles in the U.S. So when you're testing, and we'll go through how do people test, how do you make sure you don't run into problems, okay? But, and I'll say more about that. But again, the point being here is that by far most of the biocontrol agents for classical biocontrol are insects, but there are a few examples of pathogens. It'll be the total opposite for the bioherbicide approach, where it's by far mostly pathogens, very few insects. So there's a big difference between the two, and you've got all that in your notes in terms of what the differences are. Okay? One of the things I wanted to put up there, yes, Megan?
In the notes you have about the rest, it says that it could be too host specific. So is there negative implications for something being too host specific, or is that just...
Oh, I mean, it can be too host specific if by chance the biotype changes somewhat, you know, just got a little bit of a, you know, evolution, or just even just, you know, that might be you're limited. I mean, this is going to be one of the things with biocontrol. You want them to be specific, but what's the problem there by being specific? I mean, how many times do you go in an ag field and you just have velvet leaf or just ragweed? You have a combination of weeds. You might have a specific weed that's really the more problem weed. You can control the others, but you can't control this. So that's both a positive. I mean, you don't want something eating everything, but at the same time, you're limited. So sometimes if you're too host specific, means if there's any alteration in the host plant for whatever reason, that you might then, that agent might not be as effective. That's what I was trying to get at there. You know, how selective do you have to be? You know, how specific do you need to be? Be the equivalent of you know, just like in one type of apple or food item. And if something changes in it, I mean, what happens to you? I mean, you're done, right? So that's part of the problem, although we want specificity, okay? So what I'm saying here is that I'm expecting classical biological control, and I've seen this. I was asked, you know, what happens, you know, I was giving a talk to the Wheat Science Society annual meeting. They asked, you know, what do you think, Tony, in the next 5-10 years, what is going to happen to classical biocontrol? And my sense is because of the increased importance of invasive species that are non-native, example, Japanese knotweed, Japanese stiltgrass, swallowwort, kudzu, giant hogweed, a lot of species you're familiar with, and a lot of these were in what we call the lag phase. Remember, you know, invasive species, some of these plants have been here 100 years. They've kind of been under the radar for about and then all of a sudden they've spiked. And now, oh man, what are we going to do? Long term, in terms of sustainability, I mean, we can't keep spraying herbicides. I mean, firstly, it's costly. Sometimes you're just near very sensitive natural areas. You can't do it. Or near waterways, okay? And so my sense is that classical biocontrol is going to be an important long-term tool. And we see it with swallowwort, the project I'm on. That's what one of my colleague over down the road on Tower Road from the USDA, he's been hired to look at developing a biological classical control strategy against swallowwort. I mean, that's, you know, it's the first hire in 20 years that's specifically that. That tells you how serious the USDA ARS, that's Agricultural Research Service, thinks that these invasive plants are going to be. And we can't control. We don't even have herbicides to do a decent job on it. Roundup doesn't do it, okay? And the problem, of course, is going to be the non-target effects are going to be problematic, okay? in closer related species, if we get a biocontrol agent that's going to go after swallowwort, is it going to go after milkweed too, which is, they're closer related, but it's native and it's important. So, but my sense is, and you'll be hearing more about biological control going back. You're going to have, okay, so that's where I'm at. And just wanted to show you here this. Have you seen this before? Have I shown it to you in any graphs? Okay. This is a very, this is so typical of how weeds and invasive plants, their population increases over time if they have what's called the lag period. And so here's acres or area infested. At the bottom is absent or off-site. This is when the insect is introduced. This is true for an invasive plant and insect, a pathogen. Okay, this is very similar. This exponential type growth is very common. And so what I'm trying to show here is this is where if we could find a biocontrol agent or some way of controlling the plant, we should do it right here. Okay? Because look what happens. You get this exponential growth curve. Remember, that was our selected. These are our selected species. Okay? And then they'll lay off. But man, this is going to be very costly. So if you could figure out and detect early detection, rapid response is what's commonly used in the jargon, in the invasive plant and invasive insect jargon. Early detection, rapid response. You don't wait till this thing takes over everything, okay, for those that have been introduced. But what I want to show you is look at the time. By the time we or researchers notice that we have a problem, the weed has become an issue, it's way too late, okay? This is where we should be detecting, but we usually don't. So swallowwort's been around. It's been sitting there, but we haven't noticed till it's gone into exponential phase. Oh. And I wanted to, it's right here, sorry. Look at where public awareness typically begins. Okay? Right here, in the exponential phase. Okay? Eradication is out of the question by now. Okay? You need an intense, just to control this thing, let alone forget about eradication. And this is when we're noticing. We're not noticing it down here. That's why the prevention part is so important. You see something new in your own fields, in your own farm, you should jump on it. Just say, hey, I've never seen this before. You think of it as being, hey, this could be a problem. I need to look into it. If it's not, great. But very often, hey, I've got, 3/4 of my fields now have this plant. What is this thing? Should have done it much earlier. And this is where a lot of the effort from APHIS and some of the other groups, okay? So what I'm saying is that many of the species that we, have been here for 100 years are now they were in what we call the lag phase. They were just there. Maybe they needed more time to evolve or, get a greater genetic pool, adapt. And now they're at this phase. At this phase, it's game over. What we got to do now is think long-term, okay? Maybe biocontrol, sustainable. We got to think about environmental impacts. Not that some of the herbicides don't work. They work. But can we keep spraying for 20 years, 10 years? And again, in some of these areas, it's illegal to spray herbicides or chemicals because they're very sensitive. There's rare species next to them. Okay? So keep that in mind. Yes, Josh?
So in the next 50 or 100 years, do you expect that top 10 list of weeds for each state to change very much in the species composition? Do you think there are species that are lying sort of semi-dormant now that will really come through?
That's probably a certainty that that's going to happen. And we've done some projection models. There'll be a paper that will be coming out soon. I was hoping I could get it in your hands, where we've projected climate change. What's predicted, how the climate of the U.S. is going to change, particularly focusing on the Northeast, okay? And based on projections, and this is, if some of you are familiar with the Intergovernmental Panel on Climate Change, they came out with a major report, and they had what was called business as usual. If we don't do anything with our, greenhouse gases, and we don't curb fossil fuel use, it's called the, business as usual. One of the scenarios was like we really cut down like 50% of what we're doing, and another one, so do nothing and really curtail it. And we looked at those projections, and in 50 years, what we did then is looked at what the projections for the climate for New York State is going to be, precipitation-wise, and also looking at temperatures. And there's no question the climate is going to change, even if we curb, okay, what's happening now, because we're still going to be living what's happened in the 80s and 90s and 60s and 70s. It's going to happen. But So over the next 50 years, climate is going to change. And basically, I think I might have mentioned this, the climate of New York State is going to, in 40 years, current projections, and they're pretty good, it's going to resemble the climate, I believe, of Maryland. Okay? We call it the migrating states phenomena. The states migrate. And basically what we did then is looked at, say, in corn systems, what are the major weeds in Maryland? And what are the main weeds in New York State? And basically, the Northeast, relative to other regions, is going to be the region that's going to have the greatest changeover in that top 10 list. This is for agronomic weeds, let alone, invasive plants, even just agronomic weeds. Our #1 weed in New York State is going to go from being, I think we've got lamb's quarters or pigweed, one of those in there, or ragweed, to Johnson grass. Okay? Johnson grass is bad news. If any of you have tried to control it, it's a rhizomatous perennial. Okay, just sorghum alipens, which you at least you've seen some of it. Okay, so the answer is yeah, that's going to happen. So we better get used to it and understand how what are we're going to be dealing with things we've never dealt with, that we never would have thought. Okay, and of course there's other uncertainties we're just not aware of. But I would say with certainty, and there are other species that are still kind of in the lag phase. I can't tell you what are they, because geez, I wouldn't have known that swallowwort would have been there. Is it only climate? Is maybe the way we're using land? We've abandoning farms. Remember I talked about that might be a way, as in upstate New York, land was reverting back to old fields, scrub, forests, that these species took off. Because under constant cultivation, swallowwort can't handle that. It'll get chopped up. Okay? Yeah. Yeah, in the cases where they've been able to jump on it early, it's been pretty good. It's been pretty good. And I just saw that I forgot to bring in this little flyer. I just got one of our former student in the Ag Sci program down in Long Island, and some of you might have seen it. He received this from he stopped at one of these along the interstate down from Cornell. He was heading to Long Island, and this thing on Asian longhorn beetle and firewood. And I'll bring it into the IPM class. I forgot to bring it. And he said, hey, Tony, you'd love to see this because remember, you still talk about this in the class? And I said, yeah, and this whole thing of early detection and this plant being brought back and forth by firewood. And there was just a case in the news yesterday where some firewood was moving from Massachusetts to Rhode Island, and they stopped it and they found a whole bunch of Asian longhorn beetle adults in there. And oh, I mean, that's horrendous. And we talked about it. They're banning the firewood coming in from Canada, particularly if it's not treated. And so, but even here locally, they'd like you to keep wood local, not move it around. But that whole idea then is, yeah, that early detection. If you could tell folks, but it's hard. We're all like that. I mean, oh yeah, I mean, just the 1 or 2 plants, and yeah, I don't know what it is. I've never seen it before. So what I'm saying is, yeah, if that early extension education part, letting people know, hey, be on the lookout. Contact your Cornell Cooperative Extension agent or the university. Find out what it's about.
Are there any initiatives that are actually scouting for these weeds? Because one would assume that with Roundup Ready, farmers are spending less time per acre actually looking at weeds, that they're like interacting less with their fields. So is there any separate body that's doing this?
No, I mean it's the crop consultants. I try to work with them. Whenever I give talks at CCA, certified crop consultant meetings, or I talk to extension folks, I always tell them, guys, if you see something if the grower tells you or you're walking their fields, let us know. Let me know. Send me. I'll be happy to idea it. It may be nothing, but at the same time, it might be I do get that, but it's here and there. It's not. I can't say it's generalized. Somebody has to kind of really spot it. Now Cornell does have as created through New York State. Cornell there's a new institute here called the Invasive Species Institute for New York State that's housed here over in Rice Hall next door. And actually it might be a good idea. I should think about inviting Hallie Menninger, who's going to be the director, to maybe talk about that. Not just invasive plants, but invasive insects and so forth. And so they're going to be, again, with the idea that how can you coordinate? Right now we've got, I work here, there's another prof there, there's some researchers in other institutions. It'd be nice to have everybody together, and that's one of the things that they want to do, coordinate early detection. And so it might be nice to kind of bring her in and maybe just say a few words about what she's doing. So that's a reminder. But absolutely, this is when I show this, people go, yeah. I mean, we all can say, yeah. If you can, how do you do it though? That's the hard part. Okay. Yeah, Kyle.
The thing is the laws.
Right.
Our laws aren't geared toward. I mean, you can look at projections of what is more than likely going to happen. We could change the laws to make plants that are occurring in Maryland or noxious in Maryland, noxious in this state.
Right.
Because currently, we're just adding to our seedbed until the conditions are right, and then boom.
Yeah, exactly. Exactly. So right. I mean, you know, and it's very much, as we talk about in IPM, a reactive type strategy than proactive or therapeutic. We're not thinking ahead, say, hey, we have this information. I mean, we're not 100% sure, but it's likely to happen. People have told us we had Johnson grass in the weeds garden, not beyond Muncher, but on plantations where we had it previously, and they told Kathy Howard, there's no way Johnson grass is going to survive in Ithaca. It cannot survive the winters. It's 8 years we have the plant there, and if anything, it's growing bigger, and we're seeing it, and you know, we're controlling it. We don't want it to get out, but so don't listen to that.
And they found kudzu in downtown Albany that had been growing there for several years.
Yeah.
And that's an extremely southern plant, but it was
Wow.
really happy.
Right.
Going on the wall of an office building next to the people who regulate that stuff. They got it reported, and they're like, whoa.
Whoa.
It went out, and it was like, whoa, where the hell did this come from?
Kudzu, the only thing that's kept it out of New York, really, apart from this, was minimum winter temperatures. Now, as our minimum winter temperatures are changing, and you know the projections for climate change, the greatest change is not going to be, you know, really in the summer. It's a winter issue. And that's why some of you know, even when we get 20 below, oh, yeah, climate change, all right. No, that's not what they predict. We're going to have that. You're going to have these cycles, the extremes. That's what they're talking about. And everybody knows, if you look at it, there's the whole thing with lilacs. You know, lilacs are now leafing out and budding out 2 weeks earlier than in the last, you know, 50 years that they have data for. So there's no question it's happening. And also, you know, at risk. So you think about the grape industry, this things going up and down. This is the worst thing you can have for grapes. You know, coming in dormants in the middle of February or March, you know, and same for fruit trees. How many times, you know, you get your peaches because you're up to 80 degrees in April, and then boom, you get this frost, wipes everything. And we're seeing that. So the extremes is what they're talking about. Not that, so when you hear people say, oh, there's climate change. No, it's not happening. Don't, you know, you say, yeah, no, this is pretty consistent what they're saying. But those winter temperatures, so i.e. kudzu is projected to be here, really be in New York. I've seen the projections 2040, 2038 or something like that. I mean, if things go, but as we say, it's already, these things are surviving here. It's not that bad. Okay, let me just, I'll go, not all of these are relevant, not that they're not relevant. This was a presentation I had done, and I just want to talk about the bioherbicide approach. So this was the classical approach, right? The plant was non-native. We go back to the country of origin, bring it back. What plants were we targeting primarily in terms of their growth habit for classical biological control? Can you think? Because that's going to be a distinction from this bioherbicide approach. Was it annuals, high-value crops? What are we talking about? Usually they're perennials. They're usually in, you know, large areas are infested, but usually low-value areas like rangelands and so forth. Okay? Now we move to the bioherbicide approach, and it's called the bioherbicide approach because you use these products very similarly to how we use herbicides. We inundate. This is known as the inundative approach or the bioherbicide approach. Some of the biocontrol people hate the word herbicide in there. They don't like bio and herbicide. It's like some of them say, isn't that an oxymoron? You know, but often the main agent are pathogens. These are spores. This is some work I was doing up in Canada for my PhD. We're growing a fungus, the spores, and what you're seeing in the dark, that's the spores, the infective unit of this pathogen, which was called Calototricum cacoides. Okay? So I'm going to take you through some of the basics, and then I want to show you two examples. One, how this could be important in developing countries. Some of you are interested in developing countries, some of the work we had done. To the equivalent when I was in Canada of USAID. It's called the Canadian International Development Agency, CEDA. We were working in sub-Saharan Africa, in Mali in particular, trying to help the folks out with striga, witchweed. And then here at home, dandelion and turfgrass with a biocontrol agent. Okay? So the inundative approach, also referred to as the augmentative approach or the bioherbicide approach, these are all synonyms, takes advantage. Hey, who's taking plant path here? Or as a plant? The disease triangle is like kind of a basis. To get a disease or an epidemic, you need to have three things that have to work together. One, you need a host that's susceptible to the pathogen. The pathogen has to be virulent. It has to cause disease, and you need the right environmental conditions, moisture, leaf wetness, temperature. When the three work together, you get disease. This is called the disease triangle. Most often, we're trying to protect our crops, favorable plants, from getting this. What I'm talking about is actually creating the disease, because it's usually we have our host is going to be the weed, the plant we don't want, and we're trying to encourage the pathogen in the environment to be favorable for disease development. Okay? Obviously, we're not going to use a pathogen that's going to also go after our crop or neighboring plants of value. So very much the same kind of approach you need to be thinking about. The herbicide approach, you have all this information. I'll just point this out quickly. The weed is native or naturalized, just like ragweed, okay? And is therefore already in a dynamic equilibrium with its natural enemies. So the thing is, we still have the problem weed. So how come? Well, because of natural enemies and so forth, okay? Weed is controlled by manipulation of existing weed natural enemy, okay? Nearly always a plant pathogen. We're going to work with, try to mass rear, just like we were doing with the insects, but in this case, the pathogen infective unit, which in most cases could be spores, would be a good example. Okay? Large inundative population of natural enemy is applied, controls the weed, but does not survive at high levels. Control is not sustainable. Very much like a herbicide. We want to kill everything with this approach, because we know next year we're going to have to do it again. It's a bit like what we're doing with our herbicides. We have to go back next year and spray Roundup. We have to go back and spray Prowl or pendimethalin. It's different from the classical control, where we release the insects, and the hope is that the insects are going to do the job over many years. They'll, establish and keep the population below the economic threshold. Here, no, this is high-value crops. We can't wait 10 years. It's got to knock off the weed now, just like herbicides do, because otherwise competition is going to be an issue. Okay? This is referred to as, if you remember, the classical control was the ecological approach. This is often referred to as the technological approach. We're using technology. Okay? Bioherbicides are designed in the same mold as chemical herbicides, delivering transient, unsustained control. That's not to say that they don't work, but it's not sustainable over long term. Okay? I mean, if you could apply it and next year it would do the same, you could have some residual activity, but it's not high. And sometimes you don't want it to be high. And there are actually some commercial products that are available. You can go buy some products. Industry's gotten involved in this because they see a value. Okay? Not as much as we would like. Okay? These are some of the targeted weeds, just so you have an idea. Okay? This is the who's who of nasty. Hey, there's Johnson grass. If you want to know bad weeds and why USDA has targeted them, it's because they're the worst weeds we can find. Yes, Megan?
Was your example of the beetles on ragweed more of a bioherbicide then, because you had to...
Right.
It wasn't sustainable?
Yeah, that would have been more, bioherbicide, it's much closer to that. It would have been what we call, we wouldn't say bioherbicide, we call the augmentative, to augment. But it's very similar to it, okay? Even almost like a conservation. But it's much closer to this than the classical control, absolutely. So although I said it's mostly pathogens, here's an example of what we were doing that was closer to this. Okay, you don't have to remember all the names. Just to give you an idea, this is the nasties. Okay, here are some products that are out on the market. Some are still there, some are not. For those of you interested in annual bluegrass control, there's the Japanese came out with a product called Camperico. This is the trade name, okay, with the R here, trademark. A bacterial pathogen, Xanthomonas campestris, that you apply, and the bacteria will specifically knock off the annual bluegrass and not touch the turf, okay? It's been pretty good. I was trying to read the latest information on it. It is available. Fungal pathogen to control weedy tree regrowth in reforestation areas. Controlling, these are the cherries, the wild cherry species. They can be a problem, pin cherries and so forth, in forest plantations. Caligo is, this is not the pathogen I was working, it's the same genus to control a really nasty plant where they grow rice in the U.S. Where's rice in the U.S. grown? Some states, California, Mississippi, Arkansas, Louisiana. A lot of folks don't realize the U.S. is, they can produce quite a bit of rice. And Northern joint ventures is a real problem there, and they're using this fungal pathogen. And also in Florida, to control something called the vine, strangler vine. It's this vine that basically goes around the citrus plants and just over time just squeezes the heck out of them and they die. Listen to this. This is the sad part about this. Some of these, it's not the big chemical companies that get involved in this. It's usually small startup companies, biotech companies. But this product, Divine, was so efficient. Abbott Laboratories, a pretty big group, got involved in promoting this. This thing was so effective that once you applied it to the soil, okay, it basically was effective for about 7 years. So you didn't have to do it. I mean, it just kept this strangler vine, didn't allow it to germinate and so forth, didn't make it grow. It was so good that the company, the private company gave it up. It says, we can't make money out of this. This is where I think the public good has to come in. And this is what happened, is that the government came in and said, hey, we can't let this go. This is exactly, why should we have our growers? But you see what I mean? Sometimes these other products you need to apply on a regular basis, just like herbicide. You're not going to get Monsanto or Syngenta giving you a product and saying, apply it once and you're set. That's not the way business works. And we call it, you could say whatever you want, that's just the way anybody who's in business understands that profit has to be made. So here was a case where we had something really good, but no private company was going to take, well, they're going to make profit once every 7 years? No way. And so this is the public good, I call it. And you see that in biological control a lot, because often it's a niche market, but the company's not going to make money out of that, except for one product that I'll talk about, Dandelion Turf. You'll see how the private companies have jumped on the bandwagon.
Do you find any conflicting interests? Like, you were trying to get rid of ragweed, but I'm sure people that make allergy medications don't want you getting rid of ragweed.
Oh, yeah. I mean, I tried to get some funding from Merck. Merck, I mean, forget it. Burr was welcome. Now the Syngenta, the pharmaceutical, you crazy? To get $2 billion, $1.5 billion, Claritin, all of you guys that get that stuff. That's no way they were going to do. I don't think we'll ever get rid of ragweed totally. But yeah, there was no motivation for them. I was working along roadsides because in many areas, roadsides, because like they mentioned, with snow blowers and mowers that just, constantly disturb the area, many of the roadside revegetation projects fail. And I did some work looking at using other plants, clovers that are, and some grasses that are adapted to high salinity, you should see what it's like growing along a roadside, a main highway. I mean, it's gravel, it's lead, you've got all these heavy metals in there, it's no fun. But, yes, absolutely, there's going to be a conflict of interest, and so we don't even approach it. But if I'm there, I say, this is for the common good. Why should I have 10% of the population be miserable and then lose $1 billion in lost wages and time off work and so forth? Okay? What I wanted to say, and I won't cover all this, is that despite how neat this sounds, the bioherbicide approach, it hasn't been very successful. We have very few products out there available. Okay? Part of it is that it hasn't been, the private industry are not interested in very small markets. You need something big. You're not going to get Syngenta, Monsanto saying, I'm going to control velvet leaf and soybean, unless there's big money. Okay? And that was the same with the growers that you saw in that video. Why did the growers come to us for a biological control agent? Because the herbicide industry wasn't interested. Carrots and onions are still, peanuts for some than field corn or soybeans for even in Canada, in the areas I was working. So the companies weren't interested in looking at new products. In the U.S., we have what is called the IR4 program, where we actually have the government tests different herbicides on behalf of the companies. The companies won't do it themselves. They'll say, oh, there's too much, there's not enough carrots or onions being produced. We don't want to develop a new product. But let's say there's a product out there that hasn't been registered for carrots or onions, but we think that it may be good. Then there's this program, this federal program, that would pay, say, Cornell researchers to do the testing for the company. If the company usually agrees, they don't have to pay anything. And if the product is safe and works, you can register. Okay? as a minor, what's called a minor use, crop. Some of you might know that, minor use. So the company then has no liability. If the product, there's a problem with it, you can't go back to Monsanto and say, hey, you're using, the federal government assumes all liability if the product, there's a problem. But growers love it because they have an extra product that they can use, but it's, again, small market, okay? So I won't go through all of this part here, why. Part of it is industry. Part of it, we didn't do our homework in trying to team the pathogen with the target host and so forth, And also, we were trying to do it too much like a herbicide. Everything has to be, we were formulating like, oh, it's got to be sprayed like this and so forth, and that's not, and I think we know that, and we're going to be changing that, okay? And, of course, there was the whole patent issue, secrecy. It became too much big business, and not everybody was interested. So these were the constraints, and I think a lot of them we're overcoming. I mean, especially the technological is something we've got to do. So let me give you an example here. Don't worry, this is not stuff you'll be tested on. That's all the key stuff is in your notes. But let me show you this, okay? So where could the bioherbicide approach be valuable? As I see it, it would be where acceptable control of some specific weed is just not acceptable. We don't have anything in the arsenal, whether it's a herbicide or if we're working with organic growers, they don't have anything available to them to really for this nasty weed, say velvet weed. That's where we come in. Herbicides do not make sense economically or environmentally. In those situations, the bioherbicide approach might be valuable. Okay? Example, water hyacinth and striga. Okay? And you have to remember, more and more municipalities are banning, particularly in urban areas, the use of herbicides. And this is the case in Montreal, where I live, a city of 4 or 5 million people. You cannot spray pesticides any longer in the city. No matter, they don't care. Just you can't use it. You have to deal with it. So this might be a situation where now this might come in handy. And of course, now they start calling you up. Hey, do you have something to control field bindweed? Whereas in the past, you never hear it. So there has to be some pressure from the public and government. Okay? So let me just show you quickly 2 case studies. Witchweed, okay? I'll show you that in Dandelion. So for those of you working overseas or interested in international development, this was a project that I was involved in. The main culprit is this plant called Striga hermatica. It's a Scrophulariaceae, same family as the snapdragon family. Toadflax, butter and eggs, that same family. This is the plant. Beautiful, okay? When you've seen this, it's over. Your corn, it attacks all monocots, millet, rice. I mean, this is the staple foods for many parts of the world. The plant is here in the United States, in the Carolinas, okay, Georgia, but there's an eradication program, or at least quarantine. You can't move this plant around. Produces 500,000 microscopic seeds, each plant. It flowers, okay? It's parasitic, but it's a flowering plant. Basically, it does all its damage below ground. It attaches to the roots of these monocots and sucks up all the nutrients. So by the time you see the plant flowering emerge, it's already drawn, all the energies. We were working, a major biological strain in Africa, we worked with a fusarium, a fungus, Fusarium oxysporum, when I was up at the McGill lab. Okay? What this fungus did is basically cut off the communication between the plant and the seeds. So think of it as here's the roots of corn. Very often they release chemicals, exudates, things come off the root. The seed of this witchweed would detect that it'd be close to this plant and put out almost like a fungus, put out a germination tube, germinate and then attach to it. Okay? What we did is with this fungus, if we coated the seed of the crop, okay, with this fungus, the communication was broken. It was kind of a break in the communication. The seed that was sitting there did not recognize that there was the roots of this host plant next door to it. And that basically the plants didn't germinate. They did not attach to these plants. Okay, this is the fungus. Okay, so isolated. Where did we get this? We actually did surveys. We went, when they were in Africa, surveying the plants and seeing if we could find something. Very host specific to this species. Does not produce mitotoxic compounds. Anytime you're dealing with fungi and stuff, you have to be careful. Somebody, just because they're natural doesn't mean they're not, they're just safe. We've got some natural toxins that are dangerous, as more than some of our synthetics. Okay? The inoculum is produced on sorghum straw. Remember, we're in Mali, a very poor country. You can't start bringing in machinery that we have, fermentation equipment from North America, and telling them how to do it. You have to work with what they have. And so we tried to use whatever they had available, cooking ware, okay? So what we did is applied it a planting into seed pockets or as a seed dressing, and I'll show you. So we coated the fungus, the spores. We grew the fungus. We isolated it from the plant, a plant that was infected. We saw the plant. Whoa, what's killing this thing? We isolated it, looked at it, said, hey, this has good potential. And then reapplied it and did field studies both in the greenhouse and in Africa. This is some of the work we were doing there. We grind the sorghum straw, okay? We boiled, you know, we were using dirt pottery, whatever they had. We had a fermentation engineer help us, okay, microbiologist. Here are the fungus, the spores are in here, the capsules. They inoculate, okay, and then they'll pull the water out. And then the other thing about this project was that some of you have been overseas, particularly in Africa. The women do a lot of this work. They do most of this, and this was also a project that was to empower women in Mali and sub-Saharan Africa, because they did everything. Usually the males were off in the cities trying to, you know, get jobs and so forth, and all the taking care of the family and the agricultural stuff fell on them. So they were running some of these cottage industry type approaches. So what we found was there was this Arabic gum to hold. How do you hold the seed? How do you coat the seed? Just like we would do with a fungicide. Coat the seed. We used Arabic gum, something that they have. Okay? We put the powder and then we mixed it together and then coated the seeds in there and then planted the seeds. They would plant the seeds in little hills and mounds. Okay? And would grow, and the plants would do. So this coating of the fungus would grow and would protect basically the monocots, the millet and sorghum. Okay? So the impacts of this is going on right now. Immediate increase in food production and security. I mean, geez, you know, this thing is, they basically get 10% yield if the plant takes over. I mean, and it's over. It's famine. Alleviation of poverty. Again, a key here, the days where we just come in and say, hey, this is how you do it. Here's the John Deere. You know, that doesn't work in these areas. They don't have that kind. So you've got to work with what they have and be aware of their cultures. Who does what, how do they do it, and so forth. And also, what has happened is that in each of the villages that we were working in, we'd have one or two families that would be kind of the owners, in a sense, cooperative, the ones that would run the fermentation process and then give that out to the other villagers and adjoining villages. So they brought also in some income. They would trade sheep for it and so forth. It was a nice way to do it. So here's kind of success story. Okay, so kind of that's from where, and believe me, you're not going to get Syngenta or Monsanto wanting to do this, you know, because we gave the technology. I mean, the whole idea was to pass this technology to them. If it works, great. You know, you hear that sometimes of some of these multinationals, oh, we're doing this out of we're going to give this, but usually that's not a motivation. This is where the public good came in. So USAID or the equivalent up in Canada was providing us funds and the technology was transferred to them at no cost, you know, and hopefully, you know, that's going to help them. This is a different story. We're now in North America, okay? Major broadleaf weed control, urban problem, lawn care, you name it, okay? This is the standard 2,4-D, and we'll get into that next week. Environmentally favorable to do biocontrol, and because they're banning herbicides in urban areas particularly, there's a lot of motivation. Man, how many companies that are, you know, want to be green, if you could just give them a product that they could use that's reliable, they would love to do that. Well, here's an example. We call it the Dandy product. My former professor, major professor in Canada, set up his own company to develop this product. Okay, effective control of dandelion and other broadleaves, plantain, you know, Plantago lanceolata major, they're all there. This is called what's called solid substrate fermentation. You apply this as a granular, as you saw in the lab this week. Okay, this will be the dry formulation. You do it like almost you're applying fertilizer pellets. Okay, equivalent or superior. What's neat is this is the first real example that I know of, and I started, you know, was involved in the initial work that usually biocontrol agents, when you compare them to chemical control, they often don't even come, well, they don't come close. They just don't really reach that level. This product outscored our #1 chemical control, which would have been Kilex, 2,4-D, MCPA, some of our main, you know, broadleaf control in turf. Just wiped it out faster and did a number. So, I mean, I have never seen that kind of stuff. Okay? The company that Alan Watson is his name, was my major professor, if ever you want to look him up and see what he's doing. Territory Inc. is the company, so a spin-off company. He's still a professor at McGill, okay? And this is the company that he's established to move this forward. I mean, this is a campus. When I was at McGill, I just went up, a couple of years back, they banned, because Montreal banned the use of herbicides, they're on the island of Montreal. I mean, this is what you see. It's not like a Cornell. Here, they still spray. I don't know if you noticed, but they are going to spray. These guys can't. And it's like dandelion everywhere. I couldn't believe how much they had. Well, you can imagine they'd love to have a product or a homeowner. So what is this product? Okay, product description. It's a dry granular. This is what it looks like. Okay, usually it's barley grits. They grind up the barley. Okay, this is what's going to hold the fungus. And we have these grits, and we let the fungus, the mycelia of the fungus, okay, colonize it. The fungus is Sclerotinia minor. Those of you in plant pathology or have taken vegetable, do you know what this is? You should be jumping going, what? They're using Sclerotinia. This is also a major pathogen of lettuce, or it could be. But the isolate that we had found was very specific, okay? However, you have to watch it, okay? And I'll say something about that, okay? You wouldn't want to be using this if you're growing, next to a large vegetable or at least lettuce producer. You'd have to be careful. The active ingredient is an indigenous, naturally occurring fungus called Sclerotinia minor, okay? Broad host range. This is the fungus. Lettuce and peanuts primary hosts. So certainly you can't do it if you're growing peanuts, you're down in Georgia. Not a good thing to use this pathogen. Distribution via direct contact, the mycelia will grow over the weeds. Okay, does not produce apothecia or ascospores. That's important because those can be the overwintering long term. So you say, well, this year I'm going to spray my turf, but next year I'm going to have my vegetable garden there. And I'm going to have lettuce. And of course, if these spores, this is like the seeds, if they survive, next year you're going to get your lettuce is going to be hammered. So you want that kind of transiency in some ways. Okay? This is just providing some information. You may not have all that information in the notes. I just added. You won't be tested on this. This is just information to be, to make current research. Okay? The fungus is grown on autoclave barley grits. The product is air dried and packaged in 15 and 50 kilo units. Remember, 1 kilo is what? 2.2 pounds. Okay? You can broadcast or spot apply it just like you would a fertilizer. Okay? That's what the fungus, when it's growing over the dandelion. Let me just show you what it looks like. You put it, and you can spot treat it. There it is. It's a little granular. Stuff's gone. I mean, the dandelions, in some of the dandelions, there was a hole. I mean, the whole taproot was gone. After 7, I couldn't believe it. I thought, this is a joke. I mean, when I first, and then, oh, no, there used to be a dandelion in there. I mean, this is gone. 14 days, and of course, it's perfect. It loves it. It's a fungus. You apply this in the fall or spring when it's the wettest, a lot of wetness, moisture. You can irrigate. It's turf grass. There's a Canadian dime. If you've ever seen them, the little sailboat around there. The schooner that they call. Spring or fall. Okay? Lesions develop and can kill and can be achieved within 7 days, about twice as fast as Kilex. This is the main competitor. Okay? So, I don't want to say much about the issues. But anyway, this is a product that's now been registered in Canada. It's, I think, just got its approval in the U.S. Watson's been speaking with Scott's and some of the big turfgrass companies to see if they can promote the product or try it out or whatever. So any of you want to know more, I can tell you. So I know you need to get going. I was just checking if there was anything else. What to expect? Okay? I think this is going to be a major, in the urban areas, bioherbicides are going to be a big deal. As more and more herbicides get banned or pesticides, just be aware of that, okay?